Assalamualaikum and good morning. Today, Group 5 will presentation about video lab report identification of immune organ in fish. Before we start our presentation, I want to introduce my member of Group 5. Number 1 is Siti Nordana binti Isnil. Number 2 is me, Elena binti Jerit Lim. Number 3 is Christina Libia Suntai anak Imai. Number 4 is Maswe Martin. Number 5 is Onicha Evie. And the last one is Daniel Phillips. They have a 6 part that we going to presentation. Number 1 is introduction, objective, material, method, result, and conclusion. Assalamualaikum and good morning. My name is Elena binti Jerilim, BS2216005 and 08. Today, I'm going to presentation about introduction. The lymphoid organ system consists of those organs which involved in defense mechanisms through generating immune cells. Lymphoid organ vary by type of fish. In the jawless fish, true lymphoid organs are absent. These fish rely on region of lymphoid tissue within other organ to produce immune cells. For example, microhage and plasma cells are produced in the artery, kidney, and some area of the gut. Cytotomes were able to respond but poorly to T-dependent and T-independent antigen. Dairy, hangfish, nor lamprey process a thymus and secondary lymphoid organ which consists of lymphohemopoietic tissue morphologically equivalent to the bone marrow. In lamprey larvae from Amagoites, the main lymphohemopoietic forces, including the triphosoly, are full of the maggot formerly described as primitive split and the neuric fluid and adipose tissue. This is from me. Thank you.
My name is Anisha Avimakis, BS2216053. In my part, I'm going to talk about our lab objective, which has 2 main objectives. The first one is to identify the immune organ function in fish by examining the 2 major immune organs, such as spleen and kidney. The second objective is to identify and analyze the immune cell, observe tissue structure, and understand the role of this organ in the fish immune system.
The material we use for this lab is fresh fish, second Microsoft, 3 Microsoft slide and carbon slip, 4 strain saline solution, 5 dissection tools, which is scissors for step and staple, 6 Microsoft glass slide and cover slip, and 7 stand, which is Ginza stand.
So next, we move on to the methodology part of this experiment. So firstly, we need to prepare a fish sample. So a fresh fish sample was obtained from our UMS crustacean hatchery, and then the fish was sedated using transmol solution. After that, the fish was euthanized by giving a clean cut to its brain, and then the fish was cleaned by using an ethanol solution. After that, the spleen and kidney was extracted and rinsed with sterile saline solution. Then we move on to the examination of the spleen tissue. So a small section of the spleen tissue was placed on a microscope slide, and then a cover slip was placed and gently pressed down to flatten the tissue. Then the tissue is observed under a microscope, and then the structure of the cells was observed. And then documentation begins by taking pictures of the sample. Next, we have the identification and removal of the kidney. So the location of kidney is located, and then a pair of forceps was used to remove the kidney from the surrounding tissue, and then the connective tissue and blood vessels were cut to detach the spleen. Next is the examination of the kidney tissue. A small section of the kidney tissue was placed under a microscope slide, and then a drop of saline solution was added to prevent the cells from dying. And then a cover slip was placed and gently pressed down to flatten the tissue. Then the tissue was observed under a microscope, and then different types of cells can be observed, including the hematopoietic cells. And then lastly, we have the documentation of the sample taken via pictures. Lastly, we have the staining of the immune cells. The thin smears of kidney and spleen tissue were prepared on a microscope slide, and then the smears were fixed with methanol solution for 2 to 3 minutes, and then the smears were stained. After that, the slides were rinsed and dried, and then the slides were observed under a microscope and different types of immune cells were identified. Lastly, the abundance of morphology of the immune cells in the kidney and spleen were described and compared with each other.
Hello, my name is Master Martin and I will be presenting the results for the identification of immune organ in fish. First off, identification. Figure 1 shows the kidney and spleen identified inside the selected fish samples of the dissection. The kidney is identified at the furthest back of the body cavity, located along the spine of the fish, in dark red color. Meanwhile, the spleen of the specimens are located behind the intestine of the specimen, near the stomach and liver, exhibiting reddish color and elongated shape. On the right, you can see figure 1 with arrows on top showing pointing to the kidney, and lower arrow pointing to the spleen. Next up is cell view. Based on figure 2, spleen tissue contains multiple red blood cells, erythrocytes, pale-colored white blood cell, leukocyte, and macrophage, basophil, neutrophils, and granulocytes, to name a few. Figure 2 shows the image of spleen cell under microscope. That is all from me. Thank you very much for listening.
Hello, I'm Siti Nordana binti Ismail BS 221605551. For conclusion, our investigation into the spleen and kidney, two pivotal immune organs in fish, has revealed a complex network of immune cells that are essential for initiating and regulating immune responses. By analyzing this organ tissue structure, we have gained a deeper understanding of their roles in pathogen defense and overall immune system functionality. This research not only contributes to our knowledge of fish biology but also has potential implications for improving fish health management in aquaculture. That's all. Thank you.